So we have got all these type of different food. So how do we test for the presence of this food? Okay, there are 4 main types. Iodine test to test for starch. Okay, so recall that starch is only found in plants. Example, things like my bread, my potato chips, okay, or just potatoes. Okay, so glucose, recall again, my glucose is a monosaccharide. Okay, so monosaccharide being that it is of a single unit by themselves. So which means this is what we know as a reducing sugar. Okay, so reducing sugars and polysaccharides, they are not the same thing. Starch is a polysaccharide, as we seen earlier on at this part here. Okay, so starch is a polysaccharide, but over here the glucose is a monosaccharide. All right. Okay, I have got proteins and I have got fats as well. So for starch itself, you will be testing using the iodine test. Okay, so you can see here in the diagram, when I place a few drops of iodine solution into the starch solution, what happens is that this brownish solution will turn a blue-black color. Okay, so similarly over here, when I have got Benedict test, okay, this is my reducing sugar test. So initially it is a blue solution. My Benedict solution here, it is blue in color, the original solution. So when I have got increasing amount of reducing sugar, so for example, if I have got 10% glucose solution versus maybe 50% glucose solution. So the one with the 10% glucose solution will have a lighter color compared to the one with a 50% glucose solution, which will change to a brick red color. Okay, so proteins, we use Benedict test. So Benedict test itself, it is made up of your sodium hydroxide and your copper sulfate solution together. Okay, so egg white contains protein. You can see that there will be a change from a blue solution to a purple color solution. Okay, so fats is the easiest, whereby let's say I will be putting fat components like a peanut. I crush the peanut and I put it into a test tube itself, and then I will add in ethanol and water. That's when I have got a milky solution that is formed at the top layer.